Hello all. Today we are going to discuss about solute transport mechanisms. Myself, Rose Gayatri Jose. We all know that solutes need to be transported. In most of the cases, the transport is in the direction of its increasing electrochemical potential coupled to the second solute in the direction of its decreasing electrochemical potential. So, we are going to discuss four types of mechanisms. They are simple diffusion, facilitated diffusion, osmosis, active transport. Simple diffusion. It is a type of passive transport. It usually occurs when the electrochemical potentials on the two sides of a permeable membrane are different. The movement of molecules occurs along the concentration gradient until the concentration of solute is uniform on either side. So here we have to concentrate upon the equation flux equal to mobility into concentration into driving force. So the flux is the number of moles of solute crossing 1 square centimeter of membrane per second. The flux from the equation is evident that is proportional to the product of the solute mobility, which measures the ease of transport, the concentration, which measures the amount of material available to participate in the process, and the driving force. When the chemical potential of the solute is the same in the two faces bounding the membrane, the solute is in equilibrium and its flux across the membrane is zero. When the concentration is different, a chemical gradient potential is developed, which is the driving force for the flux of solute. Factors that affect the process: concentration gradient, mass or size of the solute molecule, temperature, solubility, solvent density, surface area, and thickness of the biological membrane. Some common examples: oxygen and carbon dioxide dissolved in the blood is exchanged by the process of simple diffusion. In animals, the removal of waste material occurs via simple diffusion. Bacteria rely on the process of simple diffusion for the process of nutrition within the cytoplasm. So here comes our next topic, that is facilitated diffusion. It is also a passive mediated transport, the process of biological transport in which specific structural components of biological membranes interact with particular solutes or classes of solute, thus increasing the rate at which they can cross the membrane. As the movement of substance occurs in the direction of concentration gradient, no chemical energy or ATP is required. So, the lipid bilayer of plasma membrane is usually hydrophobic, thus preventing the entry of hydrophilic and high polar molecules. So, few of the hydrophilic molecules and small ones pass across the membrane utilizing concentration gradient. The channels extend across the plasma membrane, connecting the external environment to the cytosol. Here we can see the presence of channel proteins. These are the integral proteins which are present in the biological membrane that are allowing the transport of molecules across the membrane by forming a channel. Channels usually have hydrophilic domains exposed to both extracellular and intracellular matrix. Thus, the problem of hydrophobic nature of plasma membrane is solved. The channel proteins are usually gated and they allow the closing or opening of the channels based on some signals. And these signals can be electric signal or simply the binding of a molecule. Carrier proteins are usually bulky and it is unlikely that they transport solute by diffusing from one phase of the membrane to the other. These proteins bind to the specific region in the molecule, causing conformational changes, and then move the bound molecule to the interior of the cell depending on the concentration gradient. Factors influencing concentration gradient, temperature, saturation, selectivity. Examples: transport of oxygen in blood and muscles, transport of ions like sodium, potassium, and much more. Next, we are going to discuss about osmosis. Osmosis is a quite familiar topic. It is the solvent molecules move across a semi-permeable membrane towards a region of high solute concentration. It is also a type of passive transport and is directed towards the direction that tends to equalize the solute concentration across a semi-permeable membrane. It too doesn't require any sorts of energy for the process. The factors influencing are temperature, concentration gradient, solvent potential, surface area and thickness of the membrane, and the pressure. Two types of osmosis are seen: endosmosis, exosmosis. We know that during endosmosis, the movement of water into the cell, which occurs when a cell is placed in a solution having higher concentration of water than the cell. Cells usually swell up in such cases, while exosmosis deals with the movement of water out of the cell, which occurs when a cell is placed in a solution having higher concentration of solute than the cell. Usually, the cells shrink in size. Reverse osmosis and forward osmosis are also types of osmosis. In reverse osmosis, it is a method of separation that is used to force a solvent through a semi-permeable membrane, resulting in solute molecules on one side and solvent molecules on the other side. Forward osmosis. It is another variation of osmosis where the osmotic pressure gradient is used to induce the flow of water from the sample solution to separate solutes. Osmotic pressure. This the pressure applied by a hypotonic pressure that results in the movement of solvent molecules across the semi-permeable membrane. It is the minimum pressure that should be applied to the solution to prevent the inverse flow of pure solvent across the semi-permeable membrane. It actually acts as the driving force of osmosis, and the rate of osmosis increases as the osmotic pressure increases. Example: osmosis influences the shape and size of animal cells because there is no cell wall in animal cell. The root system in plants take up water from the soil via osmosis. Next, we will discuss about active transport. The molecules are usually moving from an area of low concentration to an area of high concentration. They require the use of energy, usually in the form of ATP, adenosine triphosphate. Active transport uses specific transport proteins called pump, which uses metabolic energy to move ions or molecules against their concentration gradient. This is the opposite of diffusion, and these molecules are said to flow against concentration gradient. There are two types of active transport: primary and secondary. Primary. It is also called direct active transport or uniput. It involves using energy directly to pump a solute across a membrane against its electrochemical gradient. The various kinds of pumps used in such a mode of transport are P class, F class, V class, ABC superfamily. The P, F, and V classes transport ions only, while the ABC superfamily transport small molecules too. Secondary active transport is also known as coupled transport or co-transport. Energy is used to transport molecules across a membrane. There is no direct coupling of ATP. It relies upon the electrochemical potential difference created by pumping ions in or out of the cell. The energy is derived from the potential energy of a diffused different solute running down its concentration gradient. Carrier proteins for active transport. There are three types of proteins or transporters: uniporter, symporter, antiporter. Uniporter. They carries one specific ion or molecule. Symporter carries two different ions or molecules both in the same direction. An antiporter also carries two different ions or molecules but in different direction. These three types of carrier proteins are also found in facilitated diffusion, but they do not require ATP to work in that process. Some examples of pumps for active transport are sodium potassium ATPase, hydrogen potassium ion ATPase, and much more. That's come to the end of the topic. Hope all of you have understood. Thank you.